Good morning, I'm Jay Boyle, and I'm pleased to present the AHNS Research Awards. On behalf of the American Head and Neck Society Research Committee, I'd like to thank Dr. Weimüller and Dr. Myers for this opportunity to present these research awards and grants in this forum. All of the proposals are scored and graded through the American Academy of Otolaryngology Core Study Section, and I want to thank Marion Couch and her group at the Core for doing such a great job of evaluating our research grants. The first award that we will present is the Orlando J. Ballantyne Award. Orlando J. Ballantyne was a beloved head and neck surgeon and a tireless mentor of residents at MD Anderson Cancer Center. In his memory, the foundation of the Ballantyne family created this $10,000 one-year award for the best research proposal proposed from a resident. The winner this year is from the University of Minnesota, Rolando Rosas, for his proposal entitled Differentiation of Squamous Cancers with PPAR Gamma. Is Dr. Rosas here? Congratulations. Here's your plaque. Thank you.
Thank you.
The next award is our AHNS Pilot Grant. This is a $10,000 one-year award, and this award this year goes to Daniel Schneider, also at the University of Minnesota, I might add, for his project entitled Human Papillomavirus Associated in Head and Neck Cancer. Dr. Schneider. The next AHNS Pilot Award is also a $10,000 one-year award, and this award goes to Christopher Toe at Memorial Sloan Kettering Cancer Center in New York for his project entitled Characterizing the Inhibition of Scrove by Ubiquitinated Proteins. Dr. Toe. Apparently unable to attend. Our next award is a very meaningful award. This is the American Head and Neck Surgery and the American Academy of Otolaryngology combined Young Investigator Award. This is a $40,000 award, $20,000 a year for 2 years, a very meaningful effort. And this year, this award goes to Brandon Bentz at the University of Utah, Salt Lake City for his project entitled Non-Retinoid Metabolites of Beta-Carotene and P53 in Head and Neck Cancer. There's one other slide in this group. Could you advance the next slide? Thank you very much. One other Young Investigator Award, same thing, $40,000, a very useful start for an academic career. This goes to Mark Jamison at the University of Virginia for his project entitled CABR, a novel cancer testis antigen in head and neck squamous cell carcinoma. Dr. Jameson? Apparently not here. My next great honor is to mention to the constituency very briefly this data that was recently compiled and given to me by Michael Kupferman at MD Anderson, who did a very nice job, and I appreciate his efforts. I think it's important for the American Head and Neck Society members to understand what great use our grants have been to young career investigators. Dr. Kupferman contacted 13 of our recent award winners, and he identified that 9 out of the 13 recent grant recipients had achieved additional funding in the form of 23 grants. That includes 2 R01s, a KO8, 3 FAMRI awards, 3 SPORE awards, a VA Merit Award, 5 institutional grants, and 8 others. And this is, I think, a very impressive accumulation of grants for our young surgeon scientists in the American Head and Neck Society. 10 out of the 13 were active in laboratory research, and 11 of the 13 active in clinical research. So on the research committee, we feel this is strong evidence that our AHNS grants are helping young surgeon scientists to develop careers as independent investigators in head and neck oncology, and clearly this is an important function of the AHNS and should be continued. I'd also like to thank all of those who have donated to the American Head and Neck Society Research Foundation, because these funds will sustain the research into the future. I wanted to mention very briefly the call for requests for proposals. Our 2009 grants will be giving us a very similar slate of awards. The grants are all received and processed through the American Academy of Otolaryngology at this website. The letters of intent are due Monday, December 15th, and the applications are due January 15th. So these are excellent mechanisms to launch young investigators on a career of independent investigators, and I highly encourage your participation. The pilot awards this year, we gave 3 awards, including the Ballantine. We had 14 excellent applications, and for the 2 the Young Investigator Awards, we had 9 applications for those proposals. So a very good response, and we look forward to that in the future. Finally, if I may, I'd like to introduce Dr. Lai. Dr. Lai is giving his presentation, which is the research that he has done based on his ACS and AHNS Combined Career Development Award. Dr. Lai is assistant professor at the University of Pittsburgh and will talk to us about tumor microenvironment regulation of HIF-1 in head and neck squamous cell carcinoma, and I'm sure you'll agree that this is the type of excellent research that the American Head and Neck Society enjoys funding. Stephen? This will advance, this will go back. Okay. And here's a pointer for you.
All right, thank you. I want to thank the American Head and Neck Society and the American College of Surgeons for this award. The support has been invaluable for the start of my career and also for the work that I will present today, and I appreciate the opportunity to describe that work to you briefly this morning. Solid tumors, like head and neck squamous cell carcinoma, rapidly outgrow their oxygen and nutrient supply. In doing so, hypoxic areas develop within these tumors. Interestingly, measurement of intratumoral oxygen levels in HNSCC has demonstrated worse patient outcomes with decreased intratumoral oxygen levels. One component that likely contributes to decreased local regional control and survival is the relative resistance of hypoxic areas to radiation therapy. We chose to examine HIF-1, or the hypoxia-inducible factor 1, in head and neck cancers. HIF-1 is a transcription factor that's a heterodimer. It's composed of an alpha and beta subunit. In normoxic conditions, HIF-1 alpha is constitutively synthesized, but then rapidly ubiquitinated and shunted into the proteasome degradation pathway. It is under hypoxic conditions or low oxygen levels that HIF-1 alpha is stabilized, allowed to interact with HIF-1 beta, translocate to the nucleus, and activate a hypoxia response program. And this hypoxia response program is thought to contribute to cancer disease progression and also to be involved in invasion and metastasis. I point out in this figure that as well as the sort of oxygen-dependent pathway of regulation for HIF-1, there is also oxygen-independent regulation of HIF-1 that is influenced by growth factors and inflammatory cytokines. We began our examination of HIF-1 by utilizing paired AHNS cell lines. Specifically, we have paired cell lines that are derived from the same patient. The A cell line indicates that it comes from the primary tumor, whereas the B designates that it's from metastatic tissue in lymph nodes. We verified in an in vitro invasion assay that the basal level of invasion of the B cell line is higher than the A cell line, as would be expected. And then we noted or saw a correlation or association that, in fact, the basal level of HIF-1 alpha expression in the B cell lines or the metastatic cell lines was always higher than the A cell line, as you can see here in the 22 pair and the 37 pair. And this was true not only with respect to expression of HIF-1 alpha, but also to transcriptional activity by the HIF-1 transcription factor. In order to examine or assess the role of HIF-1 then in head and neck invasion and metastasis, we employed a HIF-1 alpha mutant that activates or is stable in these cells. And by stably expressing the HIF-1 alpha mutant in these cells, we saw that there was an increase in the normoxic or basal transcriptional activity of HIF-1, as well as an increase that followed in the invasion of these cells, or the invasive activity of these cells. We then looked in an in vivo model, specifically a floor of mouth model, where athymic nude mice were injected with cells in the floor of mouth region. Primary tumors were allowed to grow for approximately 5 weeks. The animals were then sacrificed, the primary tumors were removed, and as shown in this photograph kindly provided by Dr. Steve Kim, the cervical lymph nodes could then be identified and harvested. Frozen sections and then H&E staining allowed us to actually identify metastasis of the primary tumor into these lymph nodes. So we began by looking again at the paired cell line of 22A and 22B, and in the metastatic cell line 22B, we saw a much higher metastasis rate to the lymph nodes than the 22A cells, which is expected. What we then compared was the HIF-1 mutant compared with its control or parental cell line, and in fact saw that there was a statistically significant increase in the metastasis in this model by the HIF-1 containing cell lines. So we sought to identify, if possible, a potential mechanism for this, and what we noted in the 22A and 22B cell pairing, again, is a change in the morphology of the cells. The 22B cells are less adherent, and they also have more of a spindle shape. And in fact, we were interested in epithelial mesenchymal transition, which is important in development and is becoming more important, the role that it has in cancer progression and in invasion and metastasis. Two of the markers that we're particularly interested in are E-cadherin and vimentin, and we noted again in our paired cell lines a very clear distinction between the primary and metastatic cell lines, where E-cadherin was decreased in its expression, whereas vimentin was sharply increased in its expression. We also noted that with the addition of the HIF mutant that there was a similar change in the morphology from the parental cell line, again that they were less adherent and they also had more of a fibroblast-like shape. And indeed, when we looked at the EMT markers in the HIF-1 mutant-containing cell line, we saw that E-cadherin was decreased and vimentin was increased. And furthermore, not only with the HIF-1 mutant, but with simply exposure to hypoxia, increased exposure to hypoxia decreased E-cadherin expression and increased vimentin expression. So I mentioned earlier that the HIF-1 is thought to play a role in HIF-1 and hypoxia is thought to play a role in the resistance to radiation. We thought that HIF-1 increased activity through EMT may also contribute to head and neck cancer resistance to targeted therapy, such as erlotinib. And in fact, erlotinib has been shown via EMT EMT changes have shown that cancer cells have increased resistance to erlotinib in the lung cells. And in fact, what we see here is that with the addition of the HIF-1 mutant, that at every dose there is a decreased cell killing as compared to its parental cell line. And fitting this data into a quadratic regression model, we noted that there is indeed a statistically significant difference in the resistance of the HIF-1 containing cells as compared to their parental controls. Well, we're continuing this work with particular interest in the oxygen-independent regulation of HIF-1. Specifically, we are interested in EPO or erythropoietin, and I hope to share with you some of that work in a session tomorrow on the frontiers of basic and translational research, especially with the current clinical interest in the use of erythropoiesis stimulating agents in head and neck cancer patients and other patients with solid tumors. The future of this work will look at STAT3 and its role in oxygen-independent and dependent regulation of HIF-1. We're also interested in hypoxia imaging and the potential utility of this imaging in radiation therapy planning. And we're always looking and trying to identify and develop new inhibitors to HIF-1 activity. Of course, all of this work is not possible without an excellent team working with me, and I certainly want to thank the members of my lab, the members of the Grandis lab, my colleagues at the University of Pittsburgh, and certainly my mentor, my research mentor, Dr. Jennifer Grandis. So with that, I'd also like to thank the Society for continuing to support the research efforts of its members, and I appreciate the opportunity to present my work this morning. Thank you very much.
Thank you, Stephen. I'd like to now introduce Dr. Marilyn Wong, to give the paper awards.
Thank you. I'm very pleased to present the following awards